greetings. And so today I'm going to talk about integration of light and temperature signaling in Arabidopsis. So I'm going to focus on a particular protein that is the focus of our research in my lab. And I'm going to introduce how it's involved in integrating light and temperature signaling in Arabidopsis with respect to modulating hypocotyl elongation as well as flowering initiation. And yes, some conclusions, also questions, and potentially future applications. So I'm sure you're all familiar with the fact that light is essential for energy production in plants, but also it's a very important informational signal that is very important for controlling many aspects of plant development as well as plant architecture and adaptation. And of course, light doesn't work in isolation. It works really closely with other environmental as well as endogenous stimuli in order to regulate many processes ranging from germination, deetylation, photoprotection, as well as clock entrainment, flowering initiation, and shade avoidance response. So unlike animal systems that use rhodopsin, plants can sense, can perceive light and respond to it through different families of photoreceptors. So in Arabidopsis, there's 5 families ranging from sensing UVB to red and far red light. So UV-Arrayed is the only genetically encoded UVB receptor in nature. And the blue light receptors are the cryptochromes, the phototropins, and the side loop family. And then there's 5 different red, far red light receptors, the phytochromes. So when you look at the subcellular localization and where these proteins act, with the exception of phototropins that are plasma membrane associated proteins, all the other photoreceptors are either constitutively localized in the nucleus or they translocate upon red light perception or light perception in general to initiate changes in gene expression. So obviously, all the action is happening in the nucleus, and this is the site where you have a lot of signal integration. And so this is, the place where a lot of crosstalk is happening. Now, in addition to light signaling, so sunlight is not only important for regulating photomorphogenesis and light responses in plants, but sunlight is very important for modulating temperature on Earth, and also with respect to regulating thermomorphogenesis and in general responses to changes in the environment. So we know that climate change is happening, and it's very clear based on the trajectory that climate change leads to an increase in global temperatures, which are predicted to have major effects in crop production. So temperature increase of as little as 1 degree centigrade can result in a huge reduction in crop production. Of course, this is species specific, but basically in the next few decades, we will have major issues in terms of crop production if the temperature continues to rise. So it's very important to understand how plants respond and adapt to changes in light as well as temperature. And it's not very surprising, that light and temperature sensing share a lot of protein components, and ranging from the actual perception of light and temperature to signal integration and downstream signaling components. And again, a lot of this signal integration and all these components are found in the nucleus and lead to major changes in transcriptional reprogramming, which again lead to changes in plant morphology, development, and adaptation, such as hypocotyl elongation, leaf morphology, and flowering initiation. And in particular, shade avoidance and high ambient temperature induce very similar architectural changes due to changes in gene expression that are important for adaptation. And recently, there's very elegant studies showing that the phytochrome B red far red light receptor can also act as a thermosensor. And it has been shown that desensitization or thermal reversion of phytochrome B is light as well as temperature regulated, which in turn affects changes in gene expression in response to both stimuli. Also, early flowering 3, which is a light and clock signaling component that regulates flowering and hypocotyl elongation, has also been shown to sense temperature changes. In particular, the prion-related polyketamines of L3 can act as a reversible temperature switch, which has been shown that high temperature can lead to ELF3 liquid droplet formation, which again has an effect on the activity of ELF3 and again changes in gene expression. In addition to, of course, these thermosensors that are also involved in light signaling, there's other thermosensing mechanisms involving histone variants, RNA structure, as well as alternative splicing, but I'm not going to focus on these today. So light and temperature, not just high temperature, but also low temperature, have been shown to trigger major changes in nuclear architecture, starting from histone modifications, but also gene repositioning. So in the case of light, it has been shown that light regulated loci can move from the interior to the periphery of the nucleus, and this correlates very nicely with induction of gene expression. On the other hand, very elegant studies from the Dean lab have shown that in addition again to histone modification and loop formation, gene clustering can also lead to major changes in gene expression. And in this case, it's more related to repression of gene expression of genes, obviously, that are involved in flowering regulation. So obviously, there's a lot of signal integration that is occurring in the nucleus and also lead to changes in nuclear architecture that lead to morphological changes that are important for plant adaptation and development. Now, today I'm going to focus on a protein that we believe acts as an integrator of different environmental stimuli that was originally identified by Olivier Ludet, that was a postdoc at the Corey lab. And he used QTL mapping to identify novel light signaling mutants. And from that screen, he identified a gene called tandem zinc finger +3, because in the Bay Arabidopsis ecotype, there was a premature stop codon that basically led to a shorter hypocotyl phenotype in response to particular conditions. So this became the focus of my project when I was a postdoc at the Corey lab and now in my own lab. So just an introduction of on this protein. This protein got its name based on its domain composition. So it has 2 zinc finger and a +3 domain. And you can see in yellow, there is an extensive, what we call N-terminus, that is highly enriched in intrinsically disordered regions. And towards the C-terminus, there is, in blue, you can see the 2 zinc finger domains, and then in orange is the +3 domain. Now, zinc finger domains can associate with RNA or DNA, and they're very commonly found in transcription factors, whereas the +3 domains are less characterized. They can also associate with nucleic acids, and in yeast and humans have been shown to regulate chromatin remodeling, as well as co-transcriptional splicing. In Arabidopsis, no proteins have been characterized with respect to the +3 domain yet, and including TZP, there's 5 +3 domain containing proteins in Arabidopsis. And in addition to these domains, TZP also contains different regulatory motifs, such as a sumo interacting motif and putative summoylation sites, particularly on the N-terminus. So based on its domain composition, it was predicted that TZP potentially can regulate gene expression. And we have shown that actually in vitro TZP can associate directly with single-stranded DNA, but also with RNA, and this occurs primarily through the zinc finger in the +3 domain. And also this protein, when you tag it with a fluorescent reporter, it's constitutively localized in the nucleus and forms these punctate structures that we can call nuclear foci or nuclear bodies, in particular in response to red light illumination. Now, having a closer look to these nuclear bodies, they form these donut-shaped ring structures that potentially suggest that TZP could act as a scaffold, recruiting other proteins to these subnuclear domains. We also know that these nuclear bodies, they're obviously regulated by red light, as I mentioned, but also they're disrupted by transcriptional inhibitors in particular, suggesting that these are potential sites of transcriptional regulation. Either active transcription is happening there, or potentially transcriptional regulators congregate in these subnuclear domains. Now, in terms of function, we know that TZP regulates plant architecture and developmental transitions in response to different light stimuli. So TZP can regulate gene expression in response to blue, red, and far red light. And we and others have shown that TZP can interact directly with red light receptor phytochrome B, as well as phyA, the far red light receptor, but also it can interact with different transcriptional regulators, such as transcription factor zinc finger HD10. And we've shown that in response to blue light, TZP associates with zinc finger HD10, and together they associate with promoters of light-regulated genes that are very important for growth-promoting responses, in particular hypocotyl elongation. And what is very interesting is that TZP can regulate a lot of genes that are involved in chromatin remodeling and transcriptional regulators. So there is a huge overrepresentation of genes that are involved in chromatin regulation. And this is very interesting because we've known for a long time that chromatin regulation is important for regulating gene expression in response to light, but we don't know how these chromatin regulators are regulated themselves. In addition, we know that TZP can interact with phyB in response to red light to regulate photoperiodic flowering. So together they associate on promoters that are involved in flowering initiation, such as FT. And we have shown that these two proteins are important for maintaining a basal pool of FT in order to induce flowering initiation. Now, of course, initiation of flowering is quite a complex response, and it's not only regulated by light. There's also pathways related to high temperature, the autonomous pathway, vernalization, aging, and also hormones can regulate flowering initiation. And in addition to the photoperiodic flowering, we saw that TZP can also regulate FLC. I mean, initially we used this as a potential control, but we could see that when you overexpress TZP, you have lower FLC levels, whereas in the TZP knockout, you have the opposite effect. And initially, we couldn't really explain this, but it was only when we started investigating how TZP potentially regulates flowering initiation, and we performed some ChIP-seq and RNA-seq analysis, that we could see that TZP can potentially integrate flowering pathways, as it can associate with and regulate the expression of HDA6, a histone deacetylase, and FLD, which have been shown to act as a complex in order to regulate FLC expression in Arabidopsis. So from that, it's probably the case that TZP downregulates FLC by modulating chromatin marks through HDA6 and FLD, so not directly. And actually, this is the case. This is what is happening. When we overexpress TZP, we see a reduction in H3, K4, K20, K14 acetylation on the FLC locus, and also the same for H3, K4 trimethylation. Whereas the opposite is happening in the TZP knockout, which is what we would expect. So clearly, TZP integrates different pathways at the chromatin level. Now, in addition to integrating chromatin pathways that regulate flowering initiation, TZP is known to be part of the subnuclear multiprotein complex that regulates not only photomorphogenesis, flowering, but also thermomorphogenesis. So you can see TZP is in the middle of this complex. These interactions have been shown through mass spec, but also used to hybrid studies. So we can see that many of these components have been shown to be involved in light signaling as well as thermomorphogenesis. And in particular, I'm going to focus on the potential interaction of TZP with phyB and PCH1. So PCH1 has been shown, to be a structural component of phyB nuclear bodies, and it's been shown to stabilize phytochrome B active state and regulates phyB thermal reversion, as well as P4 abundance, which is basically one of the major regulators of hypocotyl elongation in response to light and temperature in Arabidopsis. Now, we've shown through genetic crosses and also generation of transgenic plants that TZP and PCH1 potentially have synergistic roles in regulating hypocotyl elongation in long days. So here we reveal a new role for PCH1 in long days, but also for TZP, because you can see the double mutants have elongated hypocotyls in response to long days, and this is due to induction of a number of genes that have major roles in growth promotion. And also we uncovered a new role again for these two proteins, especially with the case of TZP in red light signaling. PCH1 was known to play a role in red light signaling through regulation of phyB activity. And although TZP and phyB have been previously shown to interact, there was no clear hypocotyl elongation phenotype in red light. The only phenotype was with respect to flowering initiation. So potentially these two proteins play synergistic roles in terms of regulating hypocotyl elongation in response to red light. And here you can see when you overexpress TZP in a PCH mutant background, you can partly compensate of this lack of PCH mutation. And this potentially occurs through an increase of P4 protein abundance. As you can see here again in the double mutant, there's much more P4 abundance, in particular during the day. That again is very important for regulating hypocotyl elongation. And also in response to red light, again, you can see in the TZP PCH1 double mutant, there's more P4. And again, this correlates with the elongation of hypocotyl elongation of hypocotyls. And also we could see that TZP can modulate phyB activity. Again, we had never seen a phenotype in TZP knockouts or overexpressors in response to red light. But then when you start crossing TZP with an overexpressing phytochrome B line, you can see that overexpression of phytochrome B basically leads to hyperactive responses. So you can see very, very short hypocotyls in red light, but when you overexpress phyB in a TZP knockout, you can see that you can partially affect phyB activity, and the hypocotyls are more elongated compared to the OX phyB. And we wanted to see why is this happening? And Wei Fang, a very talented PhD student in my lab, showed that TZP can actually stabilize not only native but also transgenic phyB proteins. So here you can see native phyB levels, and you can see that especially in the light, TZP knockout has much less phyB compared to wild type, whereas overexpression of TZP somehow leads to increased levels of phyB protein. And again, when in lines overexpressing phyB, when you cross them with a TZP knockout, you can see that there's much less phyB protein accumulating. And we wanted to see if this plays any role in terms of phyB nuclear body formation. And you can see that indeed there's less and smaller nuclear bodies, but of course this could be due to the lower protein levels. And we also know that phytochrome B is essential for recruiting TZP in these nuclear bodies. So potentially this interaction is mutual. Maybe they require each other in order to localize or retain these nuclear bodies. And of course, we wanted to see whether this increase in phyB protein abundance that is promoted by TZP, if potentially TZP interaction with phyB rescues it from proteasomal degradation, and we can see MG132 treatment partially rescues this phenotype that we can see in a TZP knockout. So potentially TZP and PCH1 act synergistically in order to regulate hypocotyl elongation in response to red light and long days, and through stabilizing P4 protein. But also TZP is shown that can modulate phyB activity potentially by promoting phyB protein abundance. But of course, we cannot exclude the possibility that TZP could also affect phyB activity. And this is something we're actually working on and trying to address at the moment. Now, lastly, we were interested to find out whether TZP can integrate light and temperature signaling, and since TZP co-localizes with phytochrome B, which is a thermosensor, but also with ELF3 that has recently been shown to be a thermosensor in these nuclear photobodies, we were wondering whether TZP could play a role in thermomorphogenesis. And we performed some hypocotyl assays. We know in response to high temperature, there is an elongation of hypocotyls that is, again, mediated primarily by phytochrome B, but also by L3. So in a TZP mutant, we can see that there is a hypersensitive response to high temperature, and this is the ratio. Basically the change 28 to 22 degrees, whereas overexpression of TZP leads to a hyposensitive phenotype to high temperature. And this phenotype is exaggerated in response to red light. And this is potentially due to changes in gene expression that have been associated with light and temperature mediated hypocotyl elongation. And one of the marker genes is YACA8, and you can see the same response. There is hypersensitive response in the TZP mutant and hypersensitive in the OX-TZP line. And we can see the same phenotypes with regards to flowering initiation. So high temperature usually promotes flowering, and TZP mutant shows a hypersensitive response to high temperature, whereas overexpression of TZP is hyposensitive, suggesting that TZP is potentially a negative regulator of thermomorphogenesis. So of course, currently we're doing RNA-seq analysis to identify TZP regulated genes in response to a temperature shift, and also we will perform ChIP-seq analysis of TZP associated gene targets in response to a temperature shift. And of course, we're also very interested to monitor what is the effect of temperature on TZP protein abundance, as well as localization and interacting partners. And preliminary data from our RNA-seq analysis between col0 and TZP suggests that there is an enrichment of heat responsive genes that seems to be already on in the TZP knockout in the absence of heat stimulus. So again, this is very interesting because this could have potential applications in terms of thermotolerance if indeed TZP plays a role in protective plants to high temperature. And also we know that TZP can be sumoylated, in particular in response to high temperature, and trying to understand how this process could affect the function of TZP, it's something we're currently working on. So we're hoping to use this protein as a model to understand further what happens in terms of nuclear localization, the formation of subnuclear bodies, what other components are there, why are they there, what is their function, are they specific regulatory roles of these domains, whether it's transcription, storage, desensitization, splicing, chromatin remodeling, or all of the above, and of course, what's the impact in terms of the phenotypes, whether these information can be used, again, to engineer plants that could potentially perform better in response to a changing environment. So with that, I'd like to thank primarily Anna, Elisa, Wei, and Georgia that did most of this work, and of course everyone in my lab, our collaborators, and everyone that has helped us with reagents and very helpful discussions, and of course our funding. And yes, thank you very much, everyone, for attending today.
Thank you very much, Eirini. It's nice to see the progress being made there. Every time I hear your talk, there's a few extra things to add, so which is great to see. Some lovely genetics there from Wei as well. That's really nice stuff there. So I encourage anyone to ask a question in the Q&A, or if anyone would like to just put a virtual hand up, then please do that, and I can unmute you to do that. But so if anyone has any questions for Eirini. But let me start though. I mean, I hadn't heard about this PCH1 protein before and the interaction with TZP. So what does that PCH1 do? What type of protein is it? So
right, so it's another protein that it doesn't have any specific domains. Again, there is a lot of intrinsically disordered regions, and it has been shown, it was identified based on mass spec analysis as an interactor.
Oh no.
Up and now at Danforth Center, and it has been shown that it's part of a large complex, but it can stabilize P4, and that's how it regulates hypocotyl elongation in response to short days and red light. But recently it has been shown that it can act as a structural component for phytochrome B nuclear bodies, and basically this is where it stabilizes phyB nuclear bodies, and it regulates the activity of phyB and also the photochemistry. So it seems to be possibly one of these proteins that can act as a structural component of these photobodies, and again could potentially attract other interacting partners there in order to perform this signal integration in the nucleus.
Okay, great. Okay, so a couple of questions come in. One from someone called Caroline Dean. Lovely talk, Eirini. She says the donut shapes of TZP in the confocal are interesting. Do you think it forms a ring around things? Thanks.
Yeah, possibly. We haven't looked at this in much higher resolution. It is very difficult, especially with respect to finding out whether there is active transcription and whether there are sites of recruiting or accumulating different loci. So one of the hypotheses is that potentially they form through liquid-liquid phase separation. Again, this is something we're working on, but we don't have an answer yet. And potentially, because of this ring structure, these could attract, again, other proteins or loci, which would be really interesting in terms of chromatin architecture in general. But yeah, we don't know exactly what else is there inside these ring structures and around these ring structures. But yeah, and I don't think there has been any other reports with regards to photobodies and these ring structures. The only protein so far in terms of liquid-liquid phase separation and light signaling is cryptochromes, and they've shown that the role of these nuclear bodies are potentially acting as splicing sites, possibly during transcription also. So yes, I think there's more and more proteins that will be shown to form through liquid-liquid phase separation for light signaling.
Something to put some super resolution microscopy on there maybe for the future. Okay, so a couple of other questions. A question from Prakash, and he asks, have you checked the interaction between phyB TZP with the SIM mutant and the wild-type protein? He says simulation dependent interaction between phyB PIFs may be regulating their interaction. So do you have any information about that?
Yeah, well, we do have the transgenic lines, and we can see an effect in terms of function. And they don't basically complement the mutant phenotype. We haven't done any co-IPs yet with these lines, but yeah, definitely it would be very interesting, yeah, to see if this would affect interactions, and also subnuclear localization, of course.
Okay, so a question from Cleverson, and he says, hello, nice talk. And can you comment on anything about the relationship of this regulatory model with gigantia and the control of hypocotyl elongation? And because he just, by way of explanation, gigantia is also found in nuclear bodies, and maybe L3 and 4 interact with TZP. Yes,
that's true. The only other protein I have seen, yeah, co-localizing so well with these donuts is GI. We haven't made crosses between TZP and GI mutants, but yeah, this is definitely something on the to-do list. Yes, thank you.
Mm-hmm. So, a question from anonymous attendees. How does the protein Hi5 fit into all these interactions? Have you done anything with Hi5? Now, the classic light signaling protein.
Yeah, we haven't, but there is a paper published recently in Plant Cell from a lab of Zhang, and they work on TZP with respect to far red regulation through phytochrome A, and they have shown that TZP and Hi5 work together in regulating gene expression in response to far red light. And again, of course, COP1 is involved in regulating both Hi5 and TZP through this phyA mediated signaling pathway.
Mm-hmm. Okay. So a question from Gunjar. This looks quite specific, so I'm gonna have to take his word for this. So, what could be the potential reason for the slight down regulation of temperature-responsive gene expression in the TZP mutant at 32 degrees in comparison to ambient temperatures? Were they already upregulated? So, can you see the question there?
Yes. Yeah, this is something, it's very new data, and to be honest, I don't have an answer to this. Yes, I mean, it's something we need to investigate further, and we need to see exactly what these genes are. I mean, we noticed that a lot of these genes that are already up in the TZP mutant at 18, they're related to heat responses. But I'm not sure if they're heat shock factors or they're just involved, some of them are involved in chromatin remodeling in response to high temperature. But yes, this is something we need to find out what's the significance in terms of physiology and responses to temperature. But yeah, that's a good point. We don't know the answer to that.
I think